the topic, that is your plasmid isolation and purification. So before starting the lecture, we have to discuss that why this plasmid isolation and the purification are very important. See, this plasmid are what? It is nothing, it is a simply a genetic material. But this genetic material is what? It is an extra genetic material, extra hereditary genetic material. Now what we have to do, we have to isolate this plasmid, means that we have to isolate the properly isolate that extra hereditary genetic material. And after isolation, what we have to do, we have to purify it. Okay, purification means that we have to remove the impurities, present into it. Okay, we all know that everything is contained what? Everything is contained a impurities. So our plasmid, or the plasmid that we are choosing, that also contain our what? That also contain a impurities. So we have to remove that impurities, then we get what? We get a proper genetic material. So the today what we are going to discuss, we are going to discuss the step involved for the isolation of the plasmid and how we purify it. So let's start the lecture. Next slide. This diagram shows that this is a what? This is your plasmid. This is your plasmid, and this is your E. coli, and this is your host genome. So plasmid is what? Plasmid is a extra genomic material of the host cell apart from their host genome. Okay, Then after that, uh, again, this plasmid are what it that I already told you. This plasmid is serve as a vector, and this serve as a vector in a molecular cloning. Okay, and it is also used in the replication of the recombinant DNA sequences within that host organisms. Okay, then when we are transferring this plasmid in a laboratory, then we use a method that is called a transformation. Okay, so in laboratory we transfer this plasmid into a host cell. Suppose that this is your plasmid and this is your host cell. It also contain a genetic material of their own. Now what we have to do, we transfer this host, sorry, we transfer this um, plasmid into the host cell, and this method is known as a transformation. Okay, here that diagram are clearly said that this is your host cell, this is your recommended plasmid. Now what we have to do, we introduce this plasmid into the host cell. This third diagram shows that the transformation of the plasmid into the host cell, and the method that we are used here is a transformation. Okay, clear hai? Now the next is your recombinant DNA. This When we transfer the genes into the plasmid, then it is known as a recombinant DNA, and it also serve as what? Serve as a vector. Plasmic vector contain a three important part. Okay, when we have a plasmic vector, then it contain what? It contain a three important part. The first part, which shown by a yellow color, this part. This part is known as a uri. Okay. Then the second part, this blue line. This blue line is what? It is for our antibiotic resistant genes, which is also known as a selectable marker gene. For example, ampicillin resistance gene. Ampicillin resistant genes are what? It is a selectable marker gene or a antibiotic resistant gene. Then the third part is what? The third part is a foreign DNA, which can be inserted anywhere within within this region. Third one is your foreign DNA, which can be inserted anywhere within this region. That is also known as a cloning site. This green site is a third part of the plasmic vector. Then in the third, DAM H1 and ECO RI1. At least one recombinant reconnaissance site are present. Now, the isolation of the plasmid and then after that purification. This plasmid isolation and the purification of the plasmid are very essential part of the molecular biological procedure. Okay, now what is the plasmid that we are already discussed, which site are important, that are we already discussed. Now the most important part of the molecular biology procedure is what? It is a plasmid isolation and their purification, which is very important. Okay, so basically the purification of that isolation and the purification of that plasmid generally involve or generally completed in a three step. That is your, the first one is your growth of the bacterial cell. Okay, suppose that what we have to do, we have a bacteria. Now we grow this bacteria. Grow means that the number of the bacterial cell increases. That is known as a growth of the bacterial culture. Then harvesting and lysis of the bacteria. Harvesting means that the we remove the impurities from them by the process known as a centrifugation. Then after that we perform our lysing. Lysing means that the breaking of the cell membrane. Then the third step is a purification of the plasmid DNA. When we uh, when we break that cell membrane of the host cell, then what we have to do, we have to purify that plasmid DNA. Then the next is your growth curve of the bacterial cell. This growth curve of the bacterial cell involve four steps. The first one is your lag phase. Then the next one is your exponential phase. Then the third one is your stationary phase. And the last one is your death phase. Okay. Now, before discussing the growth of the bacterial culture or the growth curve, we have to discuss that how does bacteria are grow. So, for the growth and development of the bacteria, we required what? We required a media. This media is what? Media is nothing, it is a nutrient. Okay, this nutrition is required for the growth and the development of the bacteria. Okay, and that we discuss in that bacterial growth curve. Now, in the bacterial growth curve, the first step is what? It is a lag phase. Now, in a lag phase, bacterial cell only grow. Bacterial cell only grow, but their number does not increase. Okay, or you can also say that bacterial cell are preparing themselves for the reproduction or for the increase of their number. Here only the maturation of the bacterial cell occur, but their number does not occur. Now the next step is your exponential growth phase. This is also known as a log phase. Here the bacterial cell nutrient utilizes the nutrient, and with the help of these nutrients the bacterial cell reproduce themselves and increases their number. Okay, here the quantity of the nutrition are also high, due to which the number of the bacterial cell increases. Okay, so within a 20 minute the bacterial cell just become a double. Then after that, the third phase is your stationary phase. In a stationary phase, there is a limited amount of the nutrient occur, due to which the number of the cell does not increases. Here, no, sorry, not the number of the cell. Here, the number of the number of the increment in the cell and the death rate become equal, due to which the stationary phases occur. Then after that the availability of the nutrient decreases, due to which the bacteria goes to the next step, that is called a death phase. Here the death of the bacteria occur, and slightly or slow, or also you can say that slowly the number of the bacterial cell decreases. So this is about the bacterial growth curve. After the growth of the bacterial culture, the next step is your harvesting and lysing of the bacteria. This bacteria cell are recovered by the method that is known as a centrifugation method. Okay, centrifugation occur in a centrifuge in which the impurities are, suppose that this is your centrifuge tube. Now the impurities are occur at the bottom, and the supernatant we have what? We have a bacterial cell. Then lysing occur. In lysing, what we have to do, we break or dissolve the cell membrane of the bacteria. Here, the lysing of the bacteria involve many method. That is your treatment with the detergent. Either what we have to do, we treat the we treat the bacterial cell with the detergent, or either by using any alkali, organic solvent, or heat. Now, the method that we choose for the lysing are totally depend upon the three factors. The first one is the size of the plasmid. This is very important, which totally depend upon the choice of the method for the treatment of that lysing. Then the second one is your bacterial strain, which type of the bacteria we are using, and the third one is the technique which is used to the subsequently purification of that plasmid DNA. So these three method which totally depend upon that choosing of the factor. Then after that, the purification of the plasmid DNA also involve the three basic method. Okay, then after isolation, the next part is what? The next part is a purification. So for purification, we required the alkaline lysis method. Lysis in the presence Lysis in the presence of the detergent and the application of affinity matrices for the plasmid or proteins. So this is a diagrammatic method of these three steps. The first step involve the growth of the bacteria culture. Here it is what? It is a media. In media, we are adding a bacterial cell. Okay, now this bacteria are grow, and this represent a media which already contain the bacteria. Now the next step is your harvesting and the lysing of the bacteria. Here, when we get a media in which the bacteria are present, we centrifuge it, and during the centrifugation, we get what? This process is known as a harvesting, and we get as a precipitate. In precipitate, we get impurities. We remove that impurities, and supernatant contain the plasmid. Then after that, we introduce this bacterial cell into the host cell by the method known as a lysing of the bacteria. And the third step is what? It is a purification of that plasmid DNA. Okay, then when after introduction of the plasmid cell into that host cell, it will be purified. Sorry, before introducing into the host cell, it will, sorry, after introduction, it is the it should be purified. Then practical part, here aim is what? We have to isolate the pure plasmid DNA from a E. coli. Okay, what we have to do? We have to isolate the pure plasmid DNA from E. coli. Here, the bacterial cell is what? The bacterial cell is a E. coli. And in E. coli, we have to isolate the pure plasmid DNA, and the method that we are going to use is a alkaline lysis method. Okay. Now, principle in alkaline lysis method, the lysing of the cell occur by using a SDS and alkaline, and also the DNA present into it are denatured by using SDS and the alkaline pH. This SDS, what they do, they lysing the bacterial cell membrane and also denature the protein present into it. And due to the alkaline pH, the genomic DNA material denature and protein also denature. Then neutralization of the solution occur, and here there is a formation of the protein SDS complex, which precipitate in the tube, and subsequently both that complex DNA and protein, we remove it by the method that is known as the centrifugation, and in a supernet we get what we get are native plasmid molecules. Then after that, alkaline lysing purification method performing by these steps, that is your harvesting of the bacteria from the culture, suspension of the bacterial pellet in a suspension buffer, lysis of the bacteria, neutralization of the lysate, cleaning of the lysine, recover of the plasmid from the cleared lysine, washing of the plasmid DNA pellet, and the storage of the plasmid DNA. Then the result in which we mention that the concentration of the DNA in a microliter or a microgram. So this is all about the isolation of the plasmid and the purification of the plasmid. I hope you will understand, but if you have any query related to this topic, you are freely to ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com, or ask your query to the helpline number that are already mentioned in your slide, that is 7080833450. Thank you for watching. Please like, share and subscribe our channel.